Imagine for a moment a tiny, almost invisible building block, uh, smaller than a speck of dust, that can protect incredibly precious, fragile cargo.
Okay.
We're talking about life-saving medicines, maybe delicate vaccines, or even living cells, shielding them from drying out, from heat, decay, and all without needing any refrigeration.
Right. That's the key part.
And now think about those amazing, almost mythical creatures, the tardigrades, water bears.
Oh, yeah. The toughest animals on Earth.
Exactly. They have this unbelievable superpower. They can survive being completely dried out, like literally shriveling up, and then poof, they come back to life when you add water.
It's incredible stuff.
So what if we can actually make a synthetic, a human-made version of that natural superpower?
Well, it sounds like science fiction, doesn't it? But today on the deep dive, that's pretty close to what we're exploring. It's a truly groundbreaking discovery in materials science.
Okay.
Researchers have found this revolutionary new way to use tiny protein fragments, they're called peptides, to create what are basically smart particles.
Smart particles. How so?
Well, they can encapsulate and protect these delicate biomolecules we mentioned just by being dried. It makes them incredibly stable outside of a cold environment.
Wow. Okay, so where are we getting this information? What's the source for today's deep dive?
We're digging into a really fascinating research paper just published in the top journal, Nature Materials.
Nature Materials, okay? Prestigious.
Very. And this wasn't just one or two people. It was a huge collaborative effort. A big team, including lead authors like Dwanit Ardev, Salma Qasim, Maiva Kost, Leili Zhu, and Monetai Najaran.
That's quite a list.
It is. And the whole project was guided by the corresponding authors Yihi from the Advanced Science Research Center and the Graduate Center of CUNY in New York, and Rain V. Uline, also from their same institutions.
Got it. CUNY.
Yeah, and their findings really showcase this remarkably adaptable new material. It could genuinely change so much.
So our mission today is to unpack how it all works.
Exactly. We're going to pull back the curtain on how these new peptides actually function. What makes them so different, so unique compared to other materials.
And the implications, right? The real world stuff.
Absolutely. The incredible potential implications for everything from storing vital medicines, maybe think about vaccines in remote places, to preserving food, even agricultural uses.
Okay, let's dive in then. When I think about peptides, and correct me if I'm wrong, they're basically short chains of amino acids, right? Like protein building blocks.
That's exactly right. Short protein fragments.
Okay, so usually they form very specific sort of rigid structures, don't they? Like precise little Lego bricks locking together in a predictable way.
Yeah, that's the traditional view.
So what makes these new peptides so different? Why are they breaking the mold?
That's a great question, and it really gets to the heart of the innovation here. So traditional peptide materials, they often rely on these strong, rigid hydrogen bonds. And those bonds create ordered, stiff structures. You're right. Think one-dimensional fibers or maybe two-dimensional sheets, very predictable, like those Lego builds you mentioned, but also, well, pretty inflexible.
Stable, but not really adaptable.
Exactly. But the peptides in this study, and they're specifically looking at these minimalistic little sequences, just 3 amino acids like lysine, tyrosine, tryptophan, or KYW for short.
KYW, got it.
These fundamentally break those rules. Instead of forming rigid structures, they create what the authors call dynamic ensembles.
Dynamic ensembles.
Sure.
Okay. Less rigid.
Much less rigid, and it happens through weaker reversible interactions. The paper calls them multivalent side chain interactions.
Multivalent side chain interactions. Okay, that sounds a bit technical. Can you maybe break that down for us? Give us an analogy.
Sure. Think of it like this. Imagine a group of people at a party. Traditional peptides are like people standing in perfectly straight, unmoving lines, rigidly fixed.
Okay, very orderly.
Right. But these new KYW peptides, they're more like the crowd at a busy party, constantly shifting, mingling, interacting in flexible ways. They have many points of weak, reversible attraction.
So not one strong connection, but lots of weaker ones.
Exactly. Like, think Velcro instead of superglue. It's these multiple flexible connections, these multivalent side chain interactions that let them constantly rearrange and adapt to whatever's around them.
So that flexibility is the secret sauce.
It's absolutely key. It gives them these unique context-adaptive properties. It's a real fundamental shift in how we think about designing peptide materials.
That makes a lot more sense. Okay, so you've got these super flexible peptides just floating around in a solution. Now, this next part you mentioned is where it gets really interesting. What happens when you just let that solution dry out?
Right, you'd think it might just turn into a crust or something boring.
Yeah, like salt crusting up.
But it's anything but simple. What happens is this surprising, really elegant phenomenon called drying-induced sequential liquid phase separation, LLPS for short.
Liquid phase separation, like oil and water.
Exactly like oil and water separating, but happening within the peptide solution itself on a microscopic scale. As the water gradually evaporates, the solution doesn't just solidify uniformly, it spontaneously separates into these distinct liquid phases before it actually solidifies.
Oh, so it actively reorganizes itself as it dries. It's not just passively losing water.
Precisely. And this LLPS thing, it's actually a known protective strategy in biology. Nature uses it. Think about those tardigrades surviving drying out.
Ah, okay. So we're borrowing from nature's playbook here.
We are. And the outcome of this LLPS process is really remarkable. You end up with these films made of stiff, densely packed, but also porous spherical microparticles.
Porous spheres. Okay.
And here's a really surprising detail that the researchers, folks like Yihai and Rain V. Uline, noted in their Nature Materials paper. These particles are actually buoyant. They float.
They float. Well,
Because they form right at the air-water interface as the water evaporates, and tiny gas bubbles or microcavities get trapped inside them during this whole LLPS drying process. That makes them low density.
huh. Trapped air bubbles.
Yeah, and that buoyancy isn't just some weird quirk. It's actually a crucial clue about how they form and potentially how they function so well.
That's amazing. Buoyant particles forming just from drying.
Yeah.
It really makes you wonder, okay, if they're forming in this unique way, can we actually control it? Like, can we control their size or shape?
It's a question.
And then the million-dollar question, especially for something like a medicine or vaccine.
Mm.
What happens if you add water back? Do they just stay as solid particles, or could you get your stuff back out?
Yes, and yes. Control is possible, and the reversibility is frankly a game changer here. The team, and this includes researchers like Lei Xu and Paola Colon de Leon, they found they can indeed control the particle size and how uniform they are.
How?
Well, for instance, just by increasing the temperature during drying, making the water evaporate faster, they got smaller, more uniform particles.
Okay, faster drying, smaller particles. Makes sense.
And also, increasing the concentration of the buffer solution they used, that also resulted in smaller, more consistent particles. So temperature and concentration are like control knobs.
Got it. So you can tune them. Any other ways to control them?
Yeah, another really neat trick they found involved removing dissolved air from the solution before drying it. It's a process called degassing.
Degassing. Okay.
When they did that, they ended up with smaller, more perfectly spherical particles that had fewer pores inside.
Ah, so that directly proves the trapped gas theory. It makes them porous and buoyant.
Exactly. It demonstrates that the trapped gas is a major contributor, and by controlling it, you can fine-tune the particle properties.
Okay, and the reversibility. You said that's the game changer. How reversible are we talking?
This is truly what sets them apart. You know, many synthetic peptide materials, once they form a structure, especially after drying, that's it. They're locked in, often irreversibly.
Right, they form stable bonds.
Exactly. But these new particles, they are fully reversible. The paper shows it really clearly. You take the dried film, you add water, and instantly it redisperses. Everything dissolves back into solution.
Instantly. Wow.
Yeah. And then if you let that water evaporate again, the particle film just reappears. You can cycle it, drying, storage, redissolving.
That's the tardigrade superpower bit, the coming back to life.
Pretty much. This on-demand reversibility is a massive differentiator. It means you can store something dry and stable, then just add water when you need it.
Okay, this is all incredibly cool science. They're flexible, they do this weird phase separation thing when they dry, they form porous, floaty spheres, and you can bring them back just with water.
Right.
So let's connect the dots. What does this mean for us, for everyday people? What can these smart particles actually do out there in the real world?
This is where it gets incredibly exciting and potentially really impactful. The main application the study demonstrated, and this involved key work from authors like Lei Xu, Paola Colon de Leon, Maeva Cost, Tiffany Zhang, and Maria Perez Ferrero, is their amazing ability to efficiently encapsulate and protect sensitive biomolecules.
Sensitive biomolecules.
Yeah.
You mean things like proteins, enzymes,
Yeah.
maybe components of vaccines?
Precisely those kinds of things. They showed it works for several examples. They successfully encapsulated small fluorescent molecules like Alexa 488 and crystal violet. That showed the basic principle.
Okay, trapping small stuff.
But Critically, and this is the huge part for medicine and biotech, they encapsulated and preserved the function of delicate proteins.
Ah, preserving the function, that's key.
Absolutely. For example, they used enhanced green fluorescent protein, EGFP. It's a common lab tool. After being dried inside these peptide particles and stored at room temperature for 5 days, the EGFP still glowed green when they added water back.
Meaning it hadn't degraded, its structure was intact.
Exactly. It retained its fluorescence, indicating its structure was preserved and it was still functional, just as if it had been kept in a fridge.
5 days is good, but what about longer?
They tested that too. They used an enzyme called lysozyme. They stored it dry in these particles for 15 days.
15 days at room temp.
Yep. And afterwards, Lily Zhu and her colleagues used a specific test called an ELISA to check its activity. And it worked. The lysozyme maintained its enzymatic activity.
Wow. So it's not just physically trapping them, it's actively preserving their delicate biological function for quite a while without needing refrigeration. That is genuinely a big deal.
It's a huge deal, especially when you think about getting medicines or diagnostics to places without reliable electricity or refrigeration.
Definitely. How does the protection actually work? Is it just forming a shell?
It's a bit more sophisticated than just forming a passive shell. The mechanism involves those interactions we talked about earlier. First, the peptides and the payload, say, the protein they interact in solution before drying even starts. They use those non-covalent side chain interactions, the electrostatic attractions, the interactions involving those specific amino acids, lysine, tyrosine, tryptophan. They sort of gently bind to the protein.
So the complex before drying.
Exactly. This complexation happens first, effectively wrapping the payload in peptides before the liquid phase separation kicks in. Then, as those protective droplets form during LLPS, the payload is already inside, perfectly engulfed.
That's clever. It's like pre-packaging it.
Kind of, yeah. And the research also found something interesting. Proteins that had a higher overall electrical charge, whether positive or negative, actually got encapsulated more efficiently.
Right, so charge matters for grabbing onto the payload.
It seems to play a significant role, and what's really promising for actually using this technology is that the researchers didn't just do it in tiny lab vials, they explored ways to scale it up.
Oh, like how?
They showed they could get similar particle formation and successful EGFP encapsulation using simple heating, or applying a vacuum to speed up drying, or even using spray drying techniques.
Spray drying. That sounds like something you could potentially do on an industrial scale.
Exactly. That's vital if this is going to move beyond the lab bench and into real-world products.
This really connects back to that tardigrade idea, doesn't it?
Yeah.
Mimicking nature's own survival tricks.
It absolutely does. And what's truly remarkable here, something the lead authors Rain, V, Uline, and Yu, he really emphasized in the paper, is how closely the synthetic peptide system mimics protective strategies we see all over nature.
Not just tardigrades.
No, think also about how certain plant seeds can survive extreme drought, sometimes for years, and still be viable. Or even brine shrimp cysts. These natural systems often rely on that same liquid phase separation process, and also something called vitrification.
Vitrification, like turning into glass.
Essentially, yes.
Yeah.
Forming a glassy, non-crystalline solid state. Imagine sugar forming a hard, clear candy. It's not frozen, but it locks everything inside in place, protecting delicate molecules from damage without ice crystals forming. Nature uses disordered proteins and sugars to do this.
So these peptides are doing something similar, forming a protective, glassy matrix.
It seems very likely that vitrification is part of the final protective state within these dried particles. So this research provides this beautiful, bio-inspired, and importantly, energy-efficient way to achieve similar protection, but using these simple engineered peptides in a controlled manner.
It's like we're finally decoding nature's survival secrets and figuring out how to apply them ourselves.
Precisely. And the potential impact is huge. This discovery coming out in Nature Materials, it really holds immense promise for completely changing how we store and formulate sensitive biomolecules.
Like pharmaceuticals?
Pharmaceuticals, absolutely. Vaccines that don't need refrigeration, think about the impact on global health equity, eliminating the need for those expensive, often unreliable cold chains.
That alone would be revolutionary.
Totally. But also enzymes used in industry, making them stable at room temperature. Even agricultural products, maybe protecting beneficial microbes or seeds, making them last longer, reducing waste, improving access everywhere.
It really touches on so many areas. Wow. Okay, this has been a truly fascinating deep dive. Let's try and recap the main takeaways from this incredible research.
Sounds good.
Okay, first, Researchers, and we should mention the team again, Kwanadar Dave, Salma Qasim, Maeva Costa, Leu Shu, Mona Tayrani Najaran, led by Yehi and Rain V. Ulin, they've engineered these really unique, simple tripeptides.
Right. And the key is they don't form rigid structures like traditional peptides. Instead, they create these dynamic, adaptable ensembles using weak, flexible side chain interactions. It's a whole new design principle.
Second, these peptides do something amazing when you just let them dry. They spontaneously undergo liquid phase separation.
Which leads to the formation of these stiff, porous, and surprisingly buoyant microparticles. And these particles are incredibly good at encapsulating sensitive stuff like proteins.
And not just trapping them, but protecting them, preserving their actual function for extended periods, even weeks, without needing a fridge.
Exactly. That's crucial.
And finally, maybe the most amazing part. This whole process is completely reversible. You just add water, and boom, the encapsulated material instantly redisperses, ready to go.
Yep. Offering this groundbreaking, really energy efficient, and nature inspired way to store and stabilize things, it could genuinely transform multiple industries and, improve lives.
So the big picture here seems to be that this research really opens up exciting possibilities for a future where keeping delicate biological materials safe and effective is much simpler, cheaper, and way more sustainable.
Yeah, potentially freeing us from the huge costs and limitations of cold chains and complex storage, especially in resource-limited settings.
It leaves you wondering, could these smart peptide particles not only revolutionize vaccine storage in remote areas, but maybe also inspire totally new kinds of materials? Materials that can literally come back to life after facing extreme conditions, just like those incredible tardigrades.
It's a fascinating thought.
What other secrets are hidden in nature's survival toolkit that we might unlock just by understanding these tiny fundamental building blocks a bit better?